Okay, well, good morning everyone. Nice to see those who have turned up. You really know who the keen students are on Thursdays, so it's good to see you. Thanks for coming. Okay, we're continuing this excursion of oxidative stress, and remember in our first couple of lectures we looked at the basic definition of this process, how that it involves overproduction of damaging reactive oxygen species as a result largely of inefficiencies in coupling of oxygen utilisation to water formation or in respiratory chain in the mitochondria, but also immune cell activation also produces huge amounts of reactive oxygen. And then in the next lecture we saw why that's bad news. We looked at the ability of some of these reactive oxygen species to attack DNA, and we saw that the 4 different bases in DNA can undergo chemical alteration to form species that have mutagenic properties. Okay, now today I want to shift gears a little bit and talk about a topic that's particularly interesting to me, and that's the phenomenon of protein oxidation. So this, in some ways, at least in quantitative terms, is more significant than DNA oxidation, because as you can imagine, proteins carry out a lot of functions in cells. They do a lot of the work in living organisms to keep cells alive. So any damage to these sort of cellular workhorses can sometimes have significant consequences. So that's one reason why protein oxidation is particularly significant. But also there's just a lot more protein in a cell than there is DNA, so it's about 20 times more common. So in quantitative terms, if a free radical is formed, it's on probability grounds it's more likely it's going to attack a protein molecule than a DNA molecule, all other things being equal. Adding to this too is the fact that we've known for a long time that if you put animals or cells in a high oxygen environment, we sometimes see a loss of enzyme activity. So they just die, lose their activity. So things like aconitase, important mitochondrial enzyme, it goes down, it's sensitive to oxygen, but also as you get older, the levels of that enzyme decline. But the challenging thing about working in this area is that a lot of proteins don't seem to change much in an oxidative stress environment. They don't seem to undergo damage, they don't seem to lose activity. So this has made working in this field complicated, and trying to work out why it is that some proteins are quite susceptible to oxidative damage, others aren't very susceptible at all. And probably one of the factors that's involved here is for a given protein is knowing whether the critical sites in the active, or critical residues within the active site of the protein are actually exposed to free radicals. So the idea is that a lot of the critical participants in the enzymological function of, say, an enzyme, catalytic function, are buried deep within the protein deliberately, so that when they are exposed to free radicals, things like the hydroxyl radical, which is so short-lived it can't travel very far, it just lives for a nanosecond, and so on. This way of thinking, perhaps it's a lot of the damage that's sustained by a protein actually occurs on so-called surface residues, and doesn't necessarily involve some of those critical internally buried residues. Okay, so that's one of the reasons why studying protein oxidation can be challenging. You might be able to find damage to some of these external residues, but do you really know whether it's functionally significant for the protein, because the most vulnerable or critically important parts of the protein may well be buried. Another problem is that there's literally thousands, and I'm never quite sure what the current estimate is of the total number of proteins in a typical human cell, but it's in the tens of thousands, if not hundreds of thousands, that can potentially form. So all of those are going to be very different to each other in terms of their size, how big they are, where they're located in the cell, how long they stick around for, what their average lifespan is, how they're broken down, all those things vary enormously. And some proteins are present in enormous numbers, high copy number proteins, where they might represent, a few percent of all the proteins in a cell. Things like heat shock proteins can be very abundant in a lot of cells. But then there are very rare proteins, there are only a few copies, handful of copies. So you need an analytical approach that allows you to detect common damage to very common and abundant proteins, but also to detect those very rare proteins when they get damaged. And that's a challenge, because our technologies might just be measuring damage to high abundance proteins, whereas we're overlooking damage to rare, less common proteins. And another area too is that there's a lot of structural, functional and sort of physicochemical rather, diversity between proteins. Some proteins are membrane embedded, integral membrane proteins. They're very hard to get out of that membrane matrix. And so whatever method we're using to extract the proteins from a cell and a tissue, we might be getting out the easily extracted proteins and leaving some of the really important membrane proteins that are hard to extract behind. Okay, so those are just a few things that make working with protein oxidation a challenging topic. Now, what sort of damage can occur during protein damage by oxidants? Well, in some ways it's analogous to what we saw with the DNA the other day. It can occur, like DNA, proteins are long polymers, heteropolymers comprising multiple amino acids fused to each other. So you can imagine that if we cook it up with a free radical generating source, such as a hydroxyl radical generator, sometimes we might just see fragmentation of the peptide backbone. And so if you run a protein on a polyacrylamide gel, you just see smearing due to all the small fragments being formed. We've known about that for a long time. But then also people, when they did those sort of experiments by running an oxidised protein out on a gel, they also saw high mass proteins being formed, higher than the actual protein itself. So these are multiples. It might be 2 times, 3 times, 4 times, 10 times the mass of the parent protein. So this is because of cross-linking that can occur between damaged proteins during the process of oxidative stress. But the phenomenon we're particularly interested in is side chain oxidation of amino acid side chains. So this is analogous to oxidation of bases in DNA. So we're going to get different chemistry occurring on different side chains in amino acids to form different types of products. The reason this is useful is that it's given us some pretty handy tools for measuring protein oxidation in cells and tissues. Okay, so I'm not going to go through all the 20 amino acids in proteins and bore you silly with all the different chemical modifications that they can sustain, but I'll pick out about 4 or 5 that are particularly relevant, and just to highlight whether or not they're useful as biomarkers or not. But a very well-established product of oxidative damage to proteins, it can occur in terms of biomarkers as oxidation products of phenylalanine. So remember, phenylalanine is one of the simplest aromatic amino acids in proteins. And what happens when you expose them to hydroxyl radicals is you get hydroxylation on the ring of the molecule, the 6-membered aromatic ring, phenyl ring on the side chain. But if you remember your protein chemistry, you'll know that tyrosine is a natural constituent of proteins, one of the essential amino acids. But that's paratyrosine, where we're going to come back to that in a minute, where the hydroxy group's attached in a para position relative to the amino acid group. But what happens when a hydroxyl radical reacts with a phenylalanine molecule? It just randomly attacks, and so you get attack on the ortho and meta positions. So these are non-biological amino acids products. So they are handy biomarkers of oxidative protein damage. Used to get quite a lot of research in the old days because one of the classic ways of studying protein biochemistry was to cook up a protein with strong mineral acids like hydrochloric acids, cook it away at a high temperature, and it hydrolyzes the peptide bonds and liberates all the amino acids. And it turns out that when you do that, these survive that harsh chemistry. So people became quite interested in measuring them as markers of oxidative cell damage. And true enough, you can find these things in tissues in the body, suggesting that our proteins do sustain oxidative damage, but they don't seem to stick around or accumulate with age as a rule. So it seems that this sort of damage is accommodated by cells, and the normal proteolytic breakdown capabilities of cells can handle this sort of modification and replace proteins containing this sort of damage. Unlike some of the oxidative damage we'll see, it doesn't accumulate with age. Now then, we mentioned tyrosine as a product, those meta and paratyrosines, but even ortho and meta rather, are products of free radical attack, but even the naturally occurring essential amino acid paratyrosine can sustain damage by oxidative hydroxyl radicals. So in this case, this happens when you've got two tyrosine residues adjacent to each other, which is very common in proteins. You often get runs of tyrosines. And these are vulnerable to attack by hydroxyl radicals which abstract a hydrogen. Basically, that's what hydroxyl radicals like to do. They snaffle hydrogen atoms off biological targets, and that's very important in lipid peroxidation, as we'll see in our labs next week. But it also occurs on proteins. So hydroxyl radicals snaffle not just a proton, but an associated electron to form a hydroxyl radical, rather a tyrosyl radical, and when these happen simultaneously on adjacent amino acids, you get this cross-linking, rapid cross-linking occurring, and this forms a product known as dityrosine between two adjacent tyrosine molecules. So this is bad news for proteins. It's also very stable, so you can measure it in a acid hydrolysate of a protein, but also it's very fluorescent, highly fluorescent. And so this is one of the products that was early found in early studies in this field. When you damage a protein with oxidants, you often find these strange fluorescent peaks forming, and this is one of the constituents of that. Again, not a fantastic marker of protein oxidation. It was used for a while in the early days, but it doesn't get the same level of attention perhaps these days that it used to, simply because it's not very responsive to free radical production in cells. So it's not a great biomarker. Bear in mind too, I haven't really got time to talk about this, but tyrosine also undergoes other types of chemical modification during oxidative stress, and it can form chlorotyrosine and nitrotyrosine from reactions with hypochlorous acid and peroxynitrite. Remember I talked about that in my first lecture on Monday. And these are particularly important in things like inflammation-associated oxidative stress, and these are very widely used, or at least the resulting tyrosine modification products formed by these species are widely used markers in those sort of contexts for rheumatoid arthritis and that sort of thing. Okay, now we come to a particularly useful marker of protein oxidation known as carbonylation. And this is a very widely used marker of protein oxidation, and that's both a strength and a weakness of this approach. One of the strengths is that it's just a handy, sensitive marker of protein oxidation, but the weakness is that you can have multiple chemistries occurring that give rise to carbonylation. So when I say carbonylation, what do I mean? Well, it basically means that we're getting the introduction of a new carbonyl group in a protein, And the example I've shown here is where an arginine substituent undergoes hydroxyl radical attack to generate this species called glutamyl semialdehyde. And you can see the carbonyl group there. And this makes it reactive with an aldehyde reagent. And we'll come back to this later in the lecture, but there are different chemistries involved here. But the two that are most commonly used are dinitrophenylhydrazine, DNPH, and biotin hydrazide. And these are widely used nowadays to trap oxidised proteins in cells and tissues and then pull them out. You can pull them out with various recovery techniques and then analyse those proteins with mass spectrometry to see what proteins are getting damaged in a cell during oxidative stress. And we're actually going to use this sort of approach in our carbonyl trapping assay in a couple of weeks' time. And this is because, as I mentioned, one of the weaknesses of relying on carbonyl groups, well, it's a strength and a weakness, is that not only direct oxidation of amino acids by free radicals contributes to carbonylation, but also some of these aldehydes that are produced in significant quantities during lipid peroxidation, things like acrolein and 4-hydroxynonenal. When they react with proteins, they mainly form carbonyl retaining adducts. We'll talk about that in our lecture on Monday. Okay, so if you bear in mind that when you use some of these classic reagents, dinitrophenylhydrazine we'll use in our lab class, or biotin hydrazide, when you use these things to detect damaged carbonylated proteins in vivo and in cell culture experiments, as long as you remember it's a sort of a non-specific marker of protein oxidation, you might be measuring direct products of damage such as glutamyl semialdehyde, but also you're probably, in fact, often you're mainly measuring lipid peroxidation derived adducts. Okay, so as long as you remember that, you're just measuring non-specific damage, these technologies can be very useful indeed. Okay, so this is the one we're going to use in our lab class in a couple of weeks when we screen our molecules for carbonyl trapping activity. We're going to measure their abilities to block the carbonylation of a protein by acrolein. We'll generate acrolein enzymatically. And this is the chemistry we'll use to detect these sorts of carbonyl adductive proteins. You react with dinitrophenylhydrazine, an old reagent, been around for years. If you did organic chemistry as a first year, you probably used this stuff. When we did chem, they used to give us an unknown, and you had to sort of do some simple chemical reactions to work out whether it had certain functional groups. And this is one you do in chem to work out if a molecule has a carbonyl group, because it forms this coloured dinitrophenylhydrazine adduct. And the nice thing about this, there's easily available antibodies against dinitrophenyl modified groups, and so you can use them in this sort of in a cellular immunoassay to find damaged proteins. So that's what we're going to do in our labs. Now, the other interesting thing about carbonylation is that there's quite a lot of literature using this marker of protein oxidation, and I've always found this figure particularly fascinating because it was produced by a guy called Earl Startman, who's no longer with us, but he was a sort of a pioneer in protein oxidation research back in the sort of 70s and 80s and 90s. But he published a compilation of several studies which I thought was particularly interesting because, you know, we're interested in the role of oxygen derived radicals in aging. But what he did was they plotted data from a number of different papers, different biological systems, including cells of human origin, dermal fibroblasts, human lens from the eye. One of the most long-lived protein in the body are in the human lens. They don't get broken down and repaired, they just stick around for our whole lifetime. And then human brain obtained at autopsy, but also rat liver and even flies. And if you plot the carbonyl content of proteins from each of those different biological sources as a fraction of lifespan, the cool thing is that in all species, basically once you get through about half your lifespan, then you start accumulating higher levels of carbonyl groups. So that's bad news for some of us, but most of you guys are still on that side of the equation. But it does suggest that something happens as we get older. We start, as cells age, they start losing the battle against some of these endogenous oxidants and don't cope so well. Now then, to wrap up, there's also a couple of other useful markers of protein oxidation that have had a lot of attention in biochemistry in particular, and these are the two sulfur-containing amino acids. They're highly susceptible to oxidative damage. In fact, they're probably the most susceptible targets for free radicals. And the first one would be the cysteines. And if you've done biochemistry, you'll know that the ability to convert adjacent cysteines, or cysteines in close proximity to each other in a protein, or even in different proteins, can be a normal feature of cellular biochemistry. They form these disulfide bridges. But these also occur during oxidative damage by free radicals. So that's one of the mechanisms contributing to protein cross-linking, not the only one, but it's one of the mechanisms contributing to the formation of high-mass proteins and what we call agglomerates of proteins. And these are particularly obvious in ageing tissues. We get the formation of these high-mass protein complexes that comprise aggregates due to cross-linking chemistry. Now, thankfully, we're not completely at the mercy of this chemistry because we possess various reductases that are able to reduce disulfide bridges. And this is actually an important control mechanism for a lot of enzyme activities. And you'll learn about that in biochemistry, that a lot of enzymes regulate their activity via controlling the levels of disulfide bridges. How do they form? Well, it's thought that one of the major mechanisms is via sulfenic acid. And sulfenic acid just forms during this reaction of a thiolate with a hydrogen peroxide to form a sulfenic acid intermediate. And you can trap this with a compound called dimidone and other reagents. A lot of research around the world going on trying to find better trapping agents for cysteine. They're kind of tricky to work with in vivo because they're very unstable. And one of the reasons they're unstable is because they rapidly react with another thiol to form that disulfide. So that's that disulfide bridge forming via the intermediacy of an hydroxylated sulfur atom on a cysteine side chain. So if you use some of these probes like dimidone, it's an early study, one of the first studies that identified this oxidation product from Sora and Associates. You can see treating cells with hydrogen peroxide is this concentration dependent increase, and then at high levels you seem to have less detect forming, so it's very concentration dependent, probably because at higher concentrations you're getting a lot of the disulfide dragging the sulfenic acid away. Okay, so these are very handy markers of protein damage in vitro, but along with a lot of people, we've found it very hard to trap them in cells because they're just so unstable. Now the final amino acid I thought we'd talk about, because this leads us into the main topic of today's lecture and looking at the clinical application of all this in Alzheimer's disease, and that's methionine. So methionine is perhaps every bit as sensitive as cysteine to free radical attack, and in this case we get the formation of a sulfoxide, methionine sulfoxide, which can then, you see the sulphur's getting oxygenated here, and then sulphur, because of its high valency, it can even undergo further oxidation to form a disulfoxide, or what is that called, a sulphone rather, met SO2. And so these are very common and a very conspicuous feature of normal metabolism. Whenever you get oxygen being utilised in cells, you often get the formation of these methionine oxidation products. they're rapidly repaired at the level of the sort of sulfoxide here. The reductase is rapidly converted back to original methionine. But once it gets that second oxidation step, these are not repaired so well. So these can accumulate in cells and tissues during oxidative stress, and also in various diseases. And so if you look in the literature, you'll find a lot of literature showing information on the importance of this reductase repair capability, as well as the formation of these oxidation products in a whole lot of diseases. And the interesting thing is that knocking out methionine groups seems to enhance susceptibility to other protein oxidation products. And here's some studies done in yeast that I thought was quite interesting, where people have looked at the levels of carbonyl groups in methionine sulfoxide reductase deficient yeast, and you can see that spontaneously, as the yeast are allowed to age, they get an increase in carbonyl groups, but this goes up further in the those that can't repair oxidative damage to methionine. So there's sort of a cooperation, and it seems that whatever processes protect against protein carbonylation involve or require methionine, functional methionine groups. Okay, if you treat the same yeast with hydrogen peroxide, you see another dramatic increase in levels of carbonyls over controls, particularly in the reductase deficient animals. Okay, so this suggests that methionine sulfoxide reductase capability, the ability to repair methionine damage, may protect against other types of oxidative damage. And this leads us to some of the clinical relevance of all this, because that's what we're really interested in oxidative stress research, finding new things to measure in human samples to find out whether oxidative stress occurs in not only during healthy ageing but also in different types of diseases. And when you do some experiments on samples collected from Alzheimer's patients and you look at antibodies, the levels of methionine oxidation using antibodies, in this particular study, certainly got my attention when it came out, there's significant increase in damage to an immunoglobulin in Alzheimer's brains compared to controls, age-matched controls. So quite a striking effect, suggesting that these oxidised methionine groups accumulate in Alzheimer's. So this is my segue into this question of what role might protein oxidation be playing in this disease. So in our last lecture we looked at DNA damage and oxidation playing a role in cancer, but in this lecture I want to look at Alzheimer's disease, because this is a condition where it's thought that protein oxidation plays a significant role. Okay, so you've probably heard about this condition in other lectures and other courses, no doubt, but it's a really nasty disease. It's the most common form of progressive dementia. It's a shocking condition, and I first became aware of it when I was a young bloke about your age, because my uncle came down with it, and the sad thing about it, he was a very successful farmer down in the southwest, and he was very healthy, strong, trim bloke, but he lost, the lights go out, you lose your cognitive ability. And I remember going to see him with my mother once, and he couldn't remember his sister at all, didn't know who it was, and yet he looked a really healthy, fit bloke. And this is a tragic thing with this disease, because it involves this loss of cognitive functions, memory, very poor language skills, and then as it progresses, social problems and sensory motor skills accumulate as well. So it's quite a devastating condition. Lots of people affected by it, and the risk of it increases the longer you live, so it's seriously associated with population ageing. So there's a lot of research in trying to work out what can we do to slow down this disease and reverse it. What's going on in the Alzheimer's brain? Well, there's a lot of complicated neurobiology underlying this condition, but the basic feature is extensive neuronal loss associated with shrinking of a whole lot of brain regions. But there's a particular disappearance of cholinergic neurons, and the significance of that will come back towards the end of my lecture, because it's quite relevant to some of the proteins that have been found to be targets for reactive oxygen species damage. But it's thought that this reduction in cholinergic neurons, because cholinergic neurons extend throughout the brain regions and contribute to a lot of interconnections with other neural pathways. So this loss of cholinergic neurons has substantial effects on the plasticity of brain networks. In terms of the pathogenesis of it, there's a lot of inflammation going on, so-called neuroinflammation, and that's bad news from a free radical point of view, because these cells, when activated, produce significant quantities of free radicals. Now, in terms of the basic pathology of the diagnosis, there are two characteristic signs of an Alzheimer's brain: the neurofibrillary tangles, comprising a cytoskeletal protein, TAUR, which is highly phosphorylated. And then there are these extracellular plaques, so the tangles tend to be intracellular, the plaques tend to be extracellular, and these comprise amyloid, derived from enzymatic processing of amyloid precursor proteins. Now, so there we can see the basic pathological features. A lot of the early work on amyloid plaque deposits was actually done here at UWA in pathology. I think some of the most cited papers from UWA Medical School came out of Colin Masters' lab when he used to be at UWA before he headed off east. But he did some of this early work finding amyloid deposits in, or at least identifying the proteins involved in these plaques. There's been a lot of debate that still isn't completely resolved about what's the significance of the formation of these abnormal protein deposits in Alzheimer's disease. But amyloid plaques have particularly received a lot of attention simply because when you look at the brains of people with familial forms of Alzheimer's, autosomal dominant forms, you can see and look at the timing of the deposition of some of these plaques is the amyloid plaques clearly do strongly precede the onset of severe dementia and the symptoms associated with Alzheimer's by as much as 10 to 15 years. So this has focused a lot of research attention on amyloid. Tau proteins also accumulate, but there's less of a time lag. They tend to go up just as the disease is manifesting itself in cognitive deficits. But this is sort of the finding that amyloid plaques start accumulating, a decade before the disease becomes apparent, has sort of fueled a lot of hope that maybe we can intervene in these intervening years with drugs to slow down and delay the pathogenesis of the condition. Now, there is a lot of activity in this area. We've now gotten, I think there are now methods clinically for detecting amyloid plaques in living patients, which is really quite, as far as I know, that's becoming more available, which is quite a remarkable development, because in the old days you just had to study it. You could only confirm the diagnosis after death on an autopsy sample. Now, what's going on there in terms of amyloid? Well, the conventional story is that they derive from inappropriate proteolytic processing of a key protein, beta amyloid precursor protein, or APP. And this protein undergoes proteolysis, or cleavage, by these classes of enzymes known as secretases. And in particular, there's a beta and gamma form which act in concert to generate a toxic 42 amino acid fragment. And this is the one that gets exported and accumulates outside cells and starts forming these aggregates in the extracellular space of neuronal cells in the Alzheimer's brain. So there's been a lot of research in the pharmaceutical industry on all sorts of ways of trying to block Alzheimer's disease. None have worked particularly well, I think most people would think, but trying to block some of these secretase enzymes with small molecule inhibitors, you would think it might work because it might stop the production of these toxic peptides, but they haven't had much clinical impact or very positive clinical results, unfortunately. Okay, so this attracts the attention of researchers in terms of what might that peptide be doing, this small 42 amino acid toxic derivative of APP. So lots of things have been found over the years, and one of the interesting things is that if you put this 42 amino acid peptide, the amyloid peptide, on cultured neurons, it actually triggers apoptosis and necrosis, and also free radical production. So this seems to be a consequence of the formation of these toxic oligomers by these peptide fragments. They seem to trigger reactive oxygen species by mitochondria and trigger some of these deleterious events in terms of protein damage. But then there's a lot of other things contributing to oxidative stress in the Alzheimer's brain. People have found in those amyloid plaques, when they've extracted them and analysed them chemically, there's an awful lot of iron in them. And so people have wondered whether this is triggering that production of hydroxyl radicals and lipid radicals from lipid hydroperoxides. So that's one theory, but again, people thought, well, if that's the case, we can give patients chelating agents that trap iron and strip iron out of the amyloid plaques, and they haven't performed well in clinical trials either. So that theory is sort of still a bit provisional. Another source for free radicals in the Alzheimer's brain that's probably very significant quantitatively is the activation of these microglia, which have sort of immune functions and repair functions in cells, and they, when activated, they do the same thing as what immune cells in the periphery do. They make tons of superoxide radicals and nitric oxide, and this of course results in the production of this very toxic peroxynitrite product due to these rapid reactions between nitric oxide and superoxide. So that's another contributing process in protein damage. If that's not enough, people have also shown that within the mitochondrial genome of mitochondria within the Alzheimer's brain, you get a lot of accumulation of mutations, and this screws up the production of respiratory chain proteins. Remember, some of the respiratory chain proteins are encoded in nuclear DNA, others are encoded in mitochondrial DNA, and so the mitochondrial derived respiratory chain complexes can get screwed up and overproduce reactive oxygen because they're uncoupled, and so there's more free radicals produced during respiration than normal. And then also there's tons of lipid peroxidation occurring in the brains of these individuals too, and this is what I want to focus on in this lecture, the direct damage to proteins, particularly by lipid peroxidation products, but also by reactive oxygen. There's also another theory that I particularly like that's I think it's going to be interesting to see how this goes in coming years, but people have shown that if you're on a statin to lower your cholesterol, you seem to have a significant protective effect against Alzheimer's disease. Could be as much as 40 or 50%, I think some people think. And what's going on here? Well, people have suggested that maybe the reduction in cholesterol means that there's less cholesterol available to microbiota within the gut to produce products of cholesterol metabolism in the form of secondary bile acids, and these have been found to be elevated in the brains of Alzheimer's patients at autopsy. Significant increases in some of these bile acids, and there are particular types of bile acids made by gut microbiome constituents. And the interesting thing about this is bile acids are pretty toxic and they stimulate oxidative stress in cell culture. So this is another possible factor contributing to the increased oxidative stress occurring in the Alzheimer's brain, one that builds on knowledge of the importance of a healthy microbiome to brain health, which is fascinating stuff. Okay, but that's still awaiting confirmation. Now, just to wrap up the lecture then, I just want to look at how some of our insights into the knowledge of the types of chemical damage that free radicals can cause in proteins can actually help us understand some of these nasty clinical conditions. And the question here becomes, what's going on in terms of the proteins within the Alzheimer's brain in terms of oxidative damage? Is something occurring at that level that might be contributing to the neurodegeneration in these conditions? So in recent years, the availability of technologies for studying protein damage and protein oxidation has really improved, and we now have mass spectrometry and bioinformatic techniques that now allow us to look at damage to individual proteins in biological samples, and this is really cool. When we first started doing this, we really used the same sort of approaches we're going to use in our lab class in a couple of weeks when we look at the DNPH method for measuring carbonyl adducts in a target protein. But in this case, you take a brain sample rather, and homogenise it, run it out on, this is the old technique, the traditional approach. You run the proteins recovered from your Alzheimer's brain and controls on a isoelectric focusing strip, and you put this on a gel and run it in one direction, and it separates out proteins on the basis of their isoelectric point and their charge. And then you take that strip out and put it on top of or rather you run it on a strip initially, you run it on an isoelectric focusing strip, and then you take that strip and put it on top of an acrylamide gel and then run them in on an electric current, which separates them by size. So now you're separating all the proteins in the sample by isoelectric point and charge and size in the other direction. So this can separate out a lot of the proteins. And remember, I mentioned at the beginning of my lecture, the problem in studying protein damage is that there's so many proteins in cells, you want to be able to identify the critical ones. And then you can transfer the proteins onto a membrane and probe with an antibody against a damaged protein. And this is what we're going to do in our lab class too. And then you can pick it up and, if needs be, chop out the bit of the nitrocellulose membrane, send it off to a mass spectrometry lab, and they'll tell you what that protein is that got damaged. So one of these black proteins that's heavily damaged, you could send it off and get it identified. Now when people did that early on in 20 years ago now, a group in Kentucky that's done a lot of work on this is they found a couple of proteins were lighting up in Alzheimer's brain compared to controls. And these early studies identified glutamine synthase and creatine kinase as, and then this other enzyme, ubiquitin carboxy terminal hydrolase, important in proteolysis and turnover of proteins. All of them seem to be getting some damage via in the Alzheimer's brain. When they summarised the findings from a number of studies, it was interesting that the researchers found that proteins in particular with particular functions in cells were getting damaged, and the ones I wanted to highlight are energy related enzymes and also cholinergic system enzymes. So some of these proteins that are important to normal neuronal function and neurotransmitter function in cholinergic neurons are getting damaged by oxidants. Okay, so things got better once these newer technologies with that got rid of the need to do two-dimensional electrophoresis and running proteins on gels. We got rid of that when we could use some of these newer capture chemistries for pulling out oxidized proteins out of a complex mixture and then purifying them and then running them into a mass spec. And this has really advanced our capabilities. And you can see at the top here a couple of the chemistries that are being used. So here's a protein containing an oxidized threonine group, that semi-glutamyl aldehyde adduct I mentioned earlier in the lecture. And that carbonyl group can react with a hydrazine on dinitrophenylhydrazine to form this product. Or we can use a biotin functionalized probe where again we've got a hydrazine group here that reacts readily with the oxidized protein. So we can use affinity chromatography, in this case with DNPH targeting antibodies to pull off dinitrophenylhydrazine derivatized proteins, or avidin, which is a naturally occurring system that's exploited to pull out biotin tag proteins. Okay, so this is a way of pulling out all the oxidatively damaged proteins and getting rid of everything else, washing away everything else, and then just recovering proteins of interest that are damaged. And when they've done this, they've found some very interesting things that confirm some of those older approaches, but you get a larger spectrum of insight into the types of damage occurring in Alzheimer's brain. And again, there's some key proteins that are being damaged here that are important in energy turnover, including ATP synthase, of course, very important in oxidative phosphorylation and generating ATP within neurons within the brain. But then some of these other enzymes are very important in glycolysis and also in responding to oxidative or protein damage, some of these heat shock proteins down here that are being damaged, as well as some proteins that are important in neuronal function. So when you look at the combination of data that's been obtained in this sort of study, people have found that it seems to be that oxidative damage is occurring on multiple enzymes involved in glycolysis, but also in ATP synthesis in mitochondria, the oxidative phosphorylation, the ATP synthase as well. So that's pretty bad news for your poor old struggling ageing brain, because we depend on lots of ATP to keep neuronal function going, particularly critical in the brain, and these free radicals seem to be damaging multiple enzymes in glycolysis and also in mitochondrial ATP production. So this is bad news, because often cells can counteract mitochondrial damage by boosting glycolysis, but this doesn't seem to be an option in an Alzheimer's brain. So people have done studies looking at when does this damage occur in terms of clinical timing for the condition, because you can discriminate between these early form of the condition, preclinical AD, mild cognitive impairment, and then late Alzheimer's. And the interesting thing is that there's differences in timing, but ATP synthase and enolase, a critical glycolytic enzyme, these two proteins seem to be damaged early on in the pathogenesis of the disease, so they may well be playing a role in increasing susceptibility to this condition. So I think I'm out of time, but this just highlights. Now, if we had more time, we'd look at what is the damage occurring in these tissues, because the beauty of mass spectrometry is you can work out what's the chemical damage to the protein, not just the fact that it's carbonylated, but what's the mass of that fragment goes up by a certain amount based on whether there's a lipid aldehyde attached to it or whether it's a direct amino acid oxidation. And people find a role for hydroxynonenal, a lipid peroxidation product, in causing some of this damage. Carbonylation is also important, and then tyrosine nitration is also important, 3-NT nitrotyrosine. Okay, so several mechanisms are damaging, all of which involve oxidative stress, are damaging some of these critical proteins in the Alzheimer's brain. So it's sort of a bit worrying, isn't it? And one of the reasons why people are interested in this sort of oxidative stress, anti-oxidative stress research. Okay, I think I'm out of time. Thanks for your attention. And we'll see you on Monday, hopefully, and we'll talk about lipid peroxidation, which is also very interesting.